this. Awesome. So with that, I'll go ahead and get started. As I mentioned, this is the webinar for the 2025 community-wide assessment of Cryo-EM heterogeneous reconstruction algorithms, or CAHRA. And this is a multi-institute collaboration. People you can see as the panelists for this webinar as well. So Ellen Zhong's group at Princeton, from CCP-EM, Tom Burnley and Joel Greer, and then from the Flatiron Institute, myself and Pilar Cosio, with other people in our groups working to really build these data sets and assess the challenge results at the end. And then we also have some shout outs here from Bin Yu, who's helping us also with thinking about doing our proper ethical data analysis and those sorts of things. If you want to check out her work here, it's really nice. And then here we have a shout out to the past challenge members, which I'm going to start with a bit at the beginning for context as well. So as I mentioned, the structure, we're going to go through motivations and then cover each of the three data sets in more detail. And then Ellen, different people looking in charge of each of these data sets will talk about them, and then Ellen will do an outro as well. And then we'll have time for discussions and questions. So some of the motivations for this, I think it's very clear from, for example, machine learning community, benchmark data sets and challenges have really contributed a lot to this. Here I've showed some individual examples like MNIST and ImageNet, and I think particularly relevant to this community is the CASP challenge, which was really something that allowed AlphaFold to really assess their ability to correctly predict protein folds. And without this, progress is much harder to make, and these kinds of things can really drive fields forward. Another Cryo-EM challenge example, just to shout out, is the Kaggle challenge from CZII for Cryo-ET. And then we also come at this from a rich context of challenges and benchmarks in the Cryo-EM field. So from tasks such as particle picking to CTF to model building, from really the premier developers in this field. And then we're kind of starting here with our challenge, and also with Ellen, we have the CryoBench benchmark dataset for Cryo-EM heterogeneity to try and introduce these kinds of approaches to heterogeneity in Cryo-EM. And so I'll give a couple slides about the first challenge, because that's definitely been a part of our thought process throughout developing CAHRA. We announced that first challenge in June of 2023 and had submissions due on October 31st, Halloween of that same year. You can still access these data sets. And in this particular case, we had both real and synthetic data sets of the thyroglobulin molecule, which had this kind of conformational heterogeneity. And we asked for volumes and population distributions from participants in this challenge. And we had great participation in this from method developers, had over 41 submissions from 9 groups. And overall, the performance was good for a relatively small data set. So it was really a successful effort in this space. In this particular challenge, we included all participants in the paper and ended up anonymizing the results with each group labeled with an ice cream flavor in the figures. And part of this is because we were developing the validation metrics as we were going along with this first challenge. And we'll talk a little bit more about that and how that has motivated the second challenge. And we also found that this pairing of simulated and real data set was really nice. And we're continuing to do this with the second challenge, have both simulated and real data sets. We're taking all of these things that we learned and more to move forward with CAHRA. And as I said, all of this data from this first challenge is still available, and you can look at the metrics that we developed in the GitHub and check out our preprint on bioRxiv. And so that kind of gives a bit of a background of where we were when we started thinking about, okay, how can we do this a bit better with maybe some more simpler to compute metrics and transparent scoring and more clear policies around anonymization, for example. We still like having a kind of combination of real and synthetic data sets. And we also, because we have a larger team, we have 3 different challenges that we're tackling here with this with this new launch. And so I'm going to talk a little bit about this pose entanglement at the end here. And then we'll move on with Joel to the conformational heterogeneity, and then Ellen on the compositional heterogeneity. We'll learn more about each of those. And so for pose entanglement, the problem is basically that in single particle cryo-EM, when there's a non-uniform pose distribution, it's easy to confuse the results of heterogeneity analysis if you have like more, for example, here in this movie I showed, right? You could have a sphere or a cylinder if you just have this end-on view, and it's hard to differentiate what those are if you don't have kind of all the right poses in your data set. And we've seen in different talks, this shows up anecdotally by efforts with people who are developing various heterogeneity methods, but really how to assess this systematically is something we want to tackle here. And so we really want to exactly do this, assess the capacity of methods to accurately predict these state populations, even with non-uniform pose distribution. And we're asking, and we're giving in this data set a particle stack, but no poses, but we are providing defocus values, and we are providing templates for here 2 states, which we want you to give us the populations of. And what we want you to give you, as I just said, is the population of each state, and optionally, so that we can do some post-hoc analysis, the per image state and your final pose estimations for how you got these state estimations. And in one way, this is similar to the previous challenge in that we're asking for populations, but it's actually much simpler in that we're only asking for 2 discrete states. It's meant to be a pretty simple task, but I think it's a nice challenge in the field to ask. And now Joel will take over for conformational heterogeneity.
Okay, thank you very much, Sonya. So I'll talk about the details of the conformational heterogeneity datasets. So first of all, why are we releasing a challenge addressing the conformational heterogeneity? Well, we know that it's present in Cryo-EM datasets because we can observe how the resolution and the B factors locally vary in reconstructions. And what we'd like to do is to benchmark how faithfully algorithms and workflows which are designed to resolve this conformational heterogeneity are in fact able to do so. And through this, we seek to provide the community with the confidence to apply these approaches in order to extract and visualize the multiple conformations present within their data sets. So for this conformational heterogeneity challenge, we've chosen human pre-sequenced protease as the macromolecule to populate the challenge data set. So I've got an example in the bottom left here of an AlphaFold prediction of the structure, but I show the local resolution estimated from a consensus reconstruction in the bottom right, and this is the consensus reconstruction that we released as this challenge data set to give you a sense of the heterogeneity that's present. Okay, next slide, please. Okay, so to give you an idea of how this particular challenge is set out, first of all, in order to prepare it, we needed a source of conformational heterogeneity. So we utilized a molecular dynamics simulation as this source. We took conformations sampled from that in order to build our particle stack. So this is a synthetic particle stack. And it's this particle stack in combination with the associated metadata, which actually constitutes the conformational heterogeneity challenge dataset. And the task that we're setting participants for this dataset is very simple, simply to recover the conformational heterogeneity present using any approach that you wish. The key thing to note is that in contrast to the 2023 Flatiron heterogeneity challenge, we're asking participants to represent this heterogeneity as an ensemble of atomic models rather than a set of density maps. So we'll use this predicted ensemble and the ground truth ensemble in order to score the entries, and we'll score them based on how accurate the atomic models are, how similar the local flexibility in the ground truth and the predicted ensembles is, and whether the predicted ensembles faithfully recover the ground truth conformational change. Whether the predicted ensembles faithfully recover the conformational change that's present in the ground truth ensemble. Okay, next slide, please. Okay, so to give the exact details of this dataset, we provide 1.75 million particles in our particle stack, each extracted with a box size of 256 pixels, with 1 angstrom per pixel. The associated metadata star file is from a consensus reconstruction, so that contains the poses along with the per particle defocus values. And with the particle information, we also provide the FASTA sequence. So this will be important for building complete atomic models, but you may also want to utilize it elsewhere in your workflow. Okay, next slide, please. Okay. So once you've developed your algorithm or workflow in order to reconstruct the conformational heterogeneity present in the dataset, and are ready to submit your ensemble of atomic models, we ask that you carefully follow the guidelines that we've provided on this. So we ask for an ensemble containing between 10 and 100 atomic models, that each of those is in MMSIF formats, that they're aligned as you believe to be optimal, and that each atomic model is complete. That is that the whole chain has been built with all the residues being complete, and that all the heavy atoms are included. We'll remove any hydrogens, for example, before we actually evaluate entries. Be aware that we won't take into account any atomic B factors or atomic occupancies, and that every model in the in the ensemble will be weighted equally. Next slide, please. Okay, so in addition to collecting the atomic models, the atomic model ensembles, which allows us to assess entries against each other, we'd also like to use this challenge as an opportunity to collect data to facilitate metrics development by the community. So in this case, we're specifically interested in metrics for evaluating latent space representations and for evaluating density map representations. So if your workflow utilizes a latent space representation of the dataset, we ask that you also deposit that alongside it. Similarly, if your approach utilizes a reconstruction of density maps or a prediction of density maps, we ask that you also deposit these along with how these density maps correspond to the particles in the particle stack. So we provide example metadata formats to map particles to latent space coordinates and to map particles to density maps, which can be found on challenge websites. So we'll release this community dataset after the challenge closes under a CC by 4 license. Okay, next slide, please. Okay, that is it for the conformational heterogeneity challenge. So we hope that this particular challenge will inspire the community to develop new techniques, as well as to adapt and build upon existing algorithms and workflows in order to recover the conformational heterogeneity present in Cryo-EM datasets and represent it as ensembles of atomic models. And hopefully this work will aid the community in extracting more information from their Cryo-EM datasets and allow a better understanding of the conformational heterogeneity present. I'll now hand over to Ellen, who will introduce the final challenge dataset that we're releasing.
Cool. Thanks, Joel and Sonia. Right, so as Joel said, I'll briefly give an overview of the third dataset, which is designed to assess methods for tackling compositional heterogeneity, or COMPHET. Next slide. And so in practice, in a lot of real-world Cryo-EM experiments, there's not only conformational heterogeneity, there's also compositional heterogeneity. And specifically, compositional heterogeneity refers to variability in the identity or presence of different molecular species within the sample. So you don't necessarily know that your sample is the same protein complex. And so we've designed an experimental data set to test compositional heterogeneity. There's question marks representing the different complexes. So this is a mystery data set. And specifically, it contains 1.6 million particle images. You'll be given CTF parameters, but no poses. And obviously, we won't tell you the composition of the complexes within the sample. And so the task will be to identify and reconstruct to the best of your ability the different complexes that are present within this experimental data set. Next slide. Specifically, participants are asked to submit a density map corresponding to each complex, as well as half maps for assessing resolution. A metadata file, so either a star file or a CS file that contains the poses or the alignments for all the particles, the CTF parameters, and also the class assignments, so which particle goes into which class or density map for the complex. And something we're also curious about is a description of the processing workflow. So what were all the steps that were taken to process this data set that contains a mixture of different complexes into the final classes. Submissions will be scored based on the recovery of all the distinct species present in the sample, the specific class assignments, and also the quality of the final classes or the structures. So both assessed by the resolution and also the accuracy of the poses. Next slide. And like I mentioned, in practice, the kind of tackling of compositional heterogeneity often requires a very iterative process. And so something we're interested in assessing, or at least just evaluating, is workflow complexity. And so in addition to reconstruction accuracy, we're also going to ask for a detailed summary of the processing workflow. And something that we hope to highlight is, are there methods that are more efficient for tackling either methods or workflows for tackling compositional heterogeneity? And so for this data set, so this is a real data set, we encourage submissions both from kind of practitioners, if you have a kind of favorite 3D classification workflow that you want to try out, or from methods developers that are kind of tackling compositional heterogeneity. Okay, next slide. So with the 3 data sets, so people are welcome to submit to any of the 3 data sets. The conclusion of this challenge will be a conference next summer. And so this will be in the summer of 2026 in the UK. The QR code right here points to our website, which is this Notion website, which contains a lot more details. Next slide. So specifically, the website will have both the email listserv for announcements, and it will also have a discussion or link to a discussion space on GitHub. Next slide. And so if you have any questions, you feel free to reach out to the organizers. We also have this discussion page so that everyone can contribute their questions, and then everyone can kind of see the answers. And so if you have any kind of clarification questions, we encourage you to post them onto this GitHub discussions page. Cool. Next slide. Great. So hopefully that was just a quick overview of the 3 different data sets. One of the main aspects of this challenge is that there are different forms of heterogeneity, conformational, compositional. In terms of the image heterogeneity, kind of differences arise from both pose as well as conformation. And so we have this pose entanglement. And so hopefully we've been able to design this challenge that accommodates the breadth of different forms of heterogeneity. One thing that we've been thinking a lot about and working a lot the last kind of couple months is, how do we score the challenge? The website will have more information on the things that we care about in terms of the different submissions and how we want to evaluate. And something that we're still considering is, should we evaluate methods with one kind of unified single composite score, or preserve kind of multiple complementarity kind of metrics that capture different aspects of reconstruction quality, specifically heterogeneity analysis. I am told that I forgot to talk about the timeline. So the timeline of this challenge, you can find it on the website, but the data sets are already released. The website's linked to a Globus kind of entry that has all those data sets that we mentioned. And we have 2 submissions, a preliminary submission date and a final submission date. The preliminary submissions is sometime in March 14th in particular. And the idea for the preliminary submissions is that any kind of entrants kind of received by that date will receive initial feedback and evaluation. And then the final submissions close at the beginning of the summer on June 1st. The results will be announced in July for the workshop, which is, yes, not the summer of 2026, but September. So September 14th to the 16th in the UK. Okay. The website also has a link to our anonymization policy. So people are welcome to submit both or either anonymously or with their kind of group names or a team name. That's kind of up to your decision, but the top performing teams we will de-anonymize because I think that's the best, we think that's the best practice for the community. Hopefully the data formats are relatively standard, and I think unless there's anything else, that's it, and we're happy to turn it over for questions.